Today we are starting with the chapter 18, Body Fluids and Circulation. You have learned that all living cells, they have to be provided with nutrients, oxygen and other essential substances. And the waste and harmful substances, they have to be removed continuously for healthy functioning of the tissues. It is therefore essential to have efficient mechanism for the movement of these substances to the cells and from the cells. So different group of animals, they have evolved different methods for their transport, and simple organisms like sponges and coelentrates, they circulate water from their surroundings through their body cavities to facilitate the cells to exchange these substances. More complex organisms, they use special fluids within their body to transport such materials, and blood is the most common that is used, body fluid, that is most of the higher organisms including humans they have. Now another body fluid, that is lymph, also helps in the transport of certain substances. So in this chapter you will learn about the composition and properties of blood and lymph, and the mechanism of circulation of blood is also explained. Topics to be covered: blood, lymph, circulatory pathways, double circulation, regulation of cardiac activity, disorders of circulatory system. Now let's start with the first topic, that is blood. We know that blood is a special connective tissue that consisting of a fluid matrix, plasma and formed elements. Plasma. Plasma is a straw-colored viscous fluid that constitutes nearly 55% of the blood. 90 to 92% of plasma is water, and proteins contribute 6 to 8% of it. Fibrinogen, globulins, albumins, these are the major proteins. Now, fibrinogens, they are needed for clotting or coagulation of blood. Globulins, they are involved in defense mechanism of the body. And the albumins, they help in osmotic balance. Plasma also contains small amounts of minerals like sodium ions, calcium ions, magnesium ions, bicarbonate ions, as well as chloride ions. Glucose, amino acids, lipids, they are also present in the plasma, as they are always in the transit in the body. And factors for coagulation or clotting of blood are also present in the plasma, that is in an inactive form. Plasma without the clotting factor is called as serum. Now let's see what are the formed elements. Now formed elements, we know that erythrocytes, leukocytes and platelets, they are collectively called as formed elements, and they constitute nearly 45% of the blood. Erythrocytes, when we talk about, you can see the picture here. Erythrocytes or red blood cells, they are the most abundant of all the cells. You can see here, they are the most abundant of all the cells in blood, and a healthy adult man on an average 5 millions to 5.5 millions of RBCs per cubic meter of blood are there. RBCs, they are formed in the red bone marrow in adults. And RBCs, they are devoid of nucleus in most of the mammals and are biconcave in shape. You can see here. And they have a red colored iron containing complex protein called hemoglobin, hence the color and name of these cells. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood, and these molecules play a significant role in the transport of respiratory gases. RBCs, they have an average lifespan of 120 days, after which they are destroyed in the spleen, which is also called as the graveyards of RBCs. So you can see here the composition of the blood here, the plasma, red blood cells as well as the white blood cells are there. Leukocytes, they are also known as white blood cells, and they are colorless due to the lack of hemoglobin, and these are nucleated. You can see the nucleus present in all of them. And they are relatively lesser in number, and they average to about 6,000 to 8,000 per cubic millimeter of blood. And leukocytes, they are generally short-lived. They have two main categories. One is granulocytes, You can see here the granules, granulocytes and agranulocytes. Neutrophils, you can see here in the figure also. Neutrophils, they are, now leukocytes, they are generally short-lived, we know that, and they have two main categories, granulocytes as well as agranulocytes. Neutrophils, eosinophils and basophils. You can see here neutrophils, eosinophils and basophils. They are granulocytes. And they are the granulocytes, while lymphocytes and monocytes, you can see here lymphocytes and monocytes, they are agranulocytes. So neutrophils are the most abundant cells. These are the neutrophils, and they are 60 to 65% of the total WPCs, and basophils are least. Basophils are 0.5 to 1% among them, and neutrophils and monocytes. Neutrophils and monocytes, 6 to 8% of phagocytic cells which destroy the foreign organisms entering the body. Basophils secrete histamine. Basophils, they secrete histamine, serotonin, heparin, etc., which are involved in the inflammatory reactions. Eosinophil, 2 to 3% resist infections and are also associated with allergic reactions. Lymphocytes, which are 20 to 25%, are of two main categories. These are B and T cells. B and T lymphocytes are responsible for the immune responses of the body. Platelets. Platelets are also called as thrombocytes and are cell fragments. They are produced from the megakaryocytes. Megakaryocytes are what? These are the special cells in the bone marrow. Blood normally contains 1,50,000 to 3,50,000 platelets per cubic millimeter, maybe more than that. Platelets are release a variety of substances, and most of which are involved in the coagulation or clotting of blood. A reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. Blood groups. As you know, blood of human beings differ in certain aspects, and though it appears to be similar, but the blood groups, they are different. Various types of grouping of bloods have been done, and two such groupings are the ABO and RH. They are widely used all over the world. So now we talk about ABO blood grouping. ABO grouping is based on the presence or absence of two surface antigens or chemicals that can induce immune response. You can see here the antigens. And here we see that A and B, Group A and Group B, you can have a look at the pictures here. These A and B, the plasma of different individuals contains two natural antibodies or proteins produced in response to the antigens. The distribution of antigens and antibodies in four groups of blood group, that is A, B, AB and O. They are shown in this table, and you probably know that during blood transfusion, any blood cannot be used, and the blood of the donor has to be carefully matched with the blood of the recipient. Therefore, any blood transfusion, to avoid severe problems of clumping, that is destruction of the RBCs. What is clumping? Destruction of the RBCs. The donor's capability is also shown here in this table. From this table, it is evident that blood group O, we see here the blood group O can be donated to persons with any other blood group. Any other blood group can receive the blood from the O. Hence, O group individuals are also called as universal donors. And the persons with AB blood group, they can accept blood from persons with AB as well as the other groups of blood. Therefore, such persons are called as universal recipients. RH grouping. Another antigen, the RH antigen, similar to the one present in the rhesus monkeys. So this RH word is derived from the word rhesus monkeys. Is observed in the surface of the RBCs of the majority of nearly 80% of humans. and such individuals are called as Rh positive. And those in which this antigen is absent, they are called as Rh negative. You can see here in the positive this antigen is present, and in Rh negative it is absent. So an Rh negative person, if exposed to Rh positive blood, will form specific antibodies against the Rh antigen. Therefore, Rh group should also be matched before transfusion. A special case of Rh incompatibility, that is mismatching, has been observed between the Rh negative blood of a pregnant mother with Rh positive blood of the fetus. Rh antigen of the fetus that do not get exposed to the Rh negative blood of the mother in the first pregnancy. You can see here in the picture that how does this Rh positive father and Rh negative mother is there, and the Rh negative mother if has an Rh positive fetus. So Rh positive blood of the fetus, Rh antigen of the fetus that do not get exposed to the Rh negative blood of the mother in the first pregnancy, as the two bloods are well separated by the placenta. But during the delivery of the first child, there is a possibility of exposure of the maternal blood to the small amounts of the Rh positive blood from the fetus, and in such cases the mother starts preparing antibodies. The mother starts preparing antibodies and against this Rh antigen of her blood, and in case of her subsequent pregnancies, the Rh antibodies from the mother can leak into the blood of the fetus and destroy the fetus. So the RBCs. So this could be fatal to the fetus or could cause severe anemia and jaundice to the baby, and this condition is called as erythroblastosis fetalis. So it's a hemolytic anemia in the fetus or neonate, as erythroblastosis is caused by transplacental transmission of maternal antibodies to fetal red blood cells, and this order usually results from incompatibility between the maternal and the fetal blood groups when Rh antigen that is tested. Coagulation of blood. You know that when you cut your finger or hurt yourself, your wound does not continue to bleed for a longer time. Usually the blood stops flowing after some time. Do you know why? Blood exhibits coagulation or clotting in response to an injury or trauma, and this is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish brown scum that is formed at the site of a cut or an injury over a period of time, and it is a clot or a coagulum. We call it as a clot or a coagulum. Formed mainly of a network of threads which are called as fibrins, in which the dead and damaged formed elements of the blood, they are trapped. Fibrins, they are formed by the conversion of inactive fibrinogens in the plasma by the enzyme thrombin. And thrombins in turn are formed from the another inactive substance present in the plasma, which is called as prothrombin. So an enzyme complex thrombokinase is required. Thrombokinase, it is an enzyme complex. Okay, that is required for all these above reactions, that is for conversions. And then this complex is formed by a series of linked enzyme reactions, which is called as a cascade process, involving a number of factors present in the plasma in an inactive state. So an injury or a trauma stimulates the platelets in the blood to release certain factors which activate the mechanism of coagulation. So certain factors released by the tissues at the site of injury also can initiate coagulation, and calcium ions also play an important role in the clotting. So these are all the steps. From platelets, prothromboplastin is released, converted into thromboplastin in the presence of calcium ions. Prothrombin through calcium ions and thromboplastin converted into thrombin. Thrombin further helps in conversion of fibrinogen into fibrin. Fibrin are the polymers, and then a clot appears where RBCs, white blood cells, and all these platelets, they get entangled into those fiber-like structures. Next topic is lymph.